So welcome everyone to Symposium 6, Impact of Light Exposure on Physical and Mental Health. Thank you all for joining us today. I want to make a few announcements before we get started and I broadly introduce the topic. If you have any problem, just remember that you can send a text message to the number provided in the chat or send an email to info@srbr.org. Also, please remember that we have the no photography or recording policy in place. If you see an icon on the slide with a green check mark, then you can take a picture. If you see an icon with a stop sign, please do not take any picture. And if you have speaker that forgot to put any sign, please don't take any picture either. So be sure to promote your science using social media with the hashtag SRBR2020. And I just got news from Katia that asked us to mention the fact that they also set up a Slack workplace for us to mingle. So over the past 150 years, the use of electrical light has increased with industrialization and urbanization, and this has benefited our society by expanding our ability to be active outside of the natural day, which increase the potential for productivity as well as leisure time. However, as we all know, too much of a good thing can become a bad thing, and the increase of electrical light at night has led to light pollution. Light pollution is not only a waste of energy, but it also broadly impacts negatively organisms. It disrupts the ecosystem, impacts agriculture by decreasing the fertility of livestock, and we now have evidence that it also negatively impacts human health. So as you can see on this picture, most organisms on Earth have evolved in a highly rhythmic environment imposed by the light-dark cycle, where natural light was a potent zeitgeber. And this led to the evolution of an endogenous oscillator, which was driven by natural light and driving behavioral and physiological outputs such that an organism would perform at the most appropriate time of the day. However, this is not the environment in which we are living anymore. And if I were to depict it on a slide, it would look much more like that, where anthropogenic and artificial light in the night environment is almost ubiquitous. And this light at night, even at low level, can impinge on the proper working of the oscillator, as well as the output to lead to broad circadian dysregulation. So how exposure to light at night affect our brain and body? There is a growing body of evidence that shows that it affects not only shift workers, but also all of us by broad circadian dysregulation, as well as disruption of the sleep-wake cycle, which lead to a decrease in time and quality of sleep. And this has broad consequences for our ability to memorize as well as to learn. There is also evidence that light at night suppress melatonin, leading to dampening rhythm of melatonin, which ultimately can lead to broad metabolic dysregulation that can lead to increased risk of obesity. Melatonin suppression at night is also known to be associated with depression, and broadly, the circadian dysregulation is also proposed to be associated with increased risk of cancer as well as reproduction-related problems. It is possible that light at night could also affect our brain and body in a clock-independent way, but because of the large impact of this light at night, there is really a critical need to understand how it affects us, how it affects human health, in order to be able to mitigate it. And so today in this symposium, we have 4 speakers. Claudia Moreno and Horacio De la Iglesia will be presenting work related to this topic in humans. Sheyum Syed will present work on color preference in Drosophila, and we'll end up the session with Diego Fernandez, who will talk about mood learning and metabolism in mice. I just want to remind the audience that there will be no question following each talk. All question will happen during the 15 minutes question and answer session following the 4 talks. And I'm asking all of you to please include to whom the question is addressed in the chat to facilitate this session.
Thank you very much. I'd like to thank first Christine for inviting me to this symposium, and also Katja for organizing an amazing meeting. She's done an outstanding job of organizing 2 meetings. She organized an in-person meeting, and when she was done about a month and a half ago, she had to start again with the online format, and so far it's been an outstanding experience. So what I wanted to tell you about, it's a story that we've been following for the last couple of years, studying sleeping human communities in association with the moon phases. And as you probably know, in the last 8 years or so, as well as Claudia, I've been interested in understanding how human circadian rhythms and sleep in particular have changed when we move from a world that looked like this to a world that looked like this. And as Claudia summarized, electric light and the access to accessible sources of light and energy in general has really dramatically changed our sleep-wake cycles. And the way we've been following this is by studying a community of historic hunter-gatherers called the Tobaquom that live in northern Argentina on the border with Paraguay. This is the region where, let me just put my laser pointer here. This is the region in South America where they live at. And essentially, you know, we have several communities of this ethnic group from Argentina and Paraguay. And they live in also exposed to different extents of access to electricity and urbanization. And this has become an excellent natural experiment because these communities are all ethnically identical pretty much. I mean, we've done some genetic tests, at least for polymorphisms, and this is very clear. And they're also socially, culturally very similar. So it's a great model in which to compare the impact of the access to electricity, controlling for many other factors. And just to give you a sense of how important this impact is, I'm going to share some data that we first gathered, you know, starting about 8 years ago, then continued by Gideon Dunster, an ex-grad student in my lab. And this is a project that it's in collaboration with Sachin Panda also. This is in a polar plot what the sleep and light exposure looks like for one of the communities. In this case, this community has no access to electricity and lives in a very rural area. And what we have here is the natural daylight in a polar plot, okay? And then what we have is, first in green is the first and third quartile and the median of when this sample of people was first exposed to 5 lux of light intensity. Remember, this is only natural light, so the only source of light here is the sunlight. Then this is the time at which they were exposed to 50 lux, and this is the time at which they were exposed to 500 lux. And you can see that, of course, it's later. And then we see at the end of the day, the last time at which they were exposed to 5, last time they were exposed to 50, and the last time they were exposed to 500. And then their sleep onset, which in this case, it's about a couple of hours after sunset. And of course, they all wake up at sunrise. They have 8 to 9 hours of solid sleep. And we confirmed this with several field trips. And just to contrast this is what happened in Seattle with our college students. Gideon has been reporting college student data for the last 4 years in all 4 seasons. And this is what you see in Seattle in the winter. This is a very short photo period with a very high latitude. And I want to call your attention to several points. The first thing is the huge variability in the level of light exposure, right? And also on sleep offset and sleep onset. But most of all, what I mainly want you to focus on is the amazing delay in this light exposure to physiological levels of light. light levels that are sufficient to stimulate the circadian system. And what's really outstanding is that indeed they are entrained to this artificially imposed light-dark cycle, because all this light can only be from artificial sources, because sunset ended here, right? But they're still going to sleep right after that exposure to the artificial light. But amazingly, we, as you all know, we're still waking up at sunrise, right? So that means that we're sleeping about half of what these people are sleeping. Not half, but we're sleeping at least 3 hours less than what the Tobacuom are sleeping in Argentina without electricity. Going back to, so this is what, the conclusion that we drew from several years of work. This started in 2012, that, the mere access to electricity leads to later sleep onsets, duration of sleep that it's about an hour shorter in the same communities, but that either have or do not have access to electricity. Increased variability both within and between individuals of their sleep timing, and a delayed phase of the circadian system. We've shown this by measuring the daily melatonin onset, that indeed the communities that are exposed to electricity have a delayed onset. So looking at this data, we should remind the notion of chronotype, and Claudia's talk was a great introduction to this. And this is, from Thiel's latest article, sort of criticizing a little bit the notion of chronotype, which he indeed pioneered. And in fact, I think that we need to be very serious about that criticism, and I'd like to push it even further. When you look at this plot that I showed you in the first slide, really, when you look at this sleep timing, you realize that there's virtually no variation of chronotype whatsoever. Claudia mentioned this also in her communities. Ken Wright has shown this in his own lab by taking them out camping. And essentially, what emerges from this is that chronotype is only enabled by the access to electricity or the access to artificial light, and particularly in highly urbanized societies. And that may be an artifact, a phenotype that only can emerge under those conditions. That's one possibility. The other possibility is that there may be in nature real selective pressure for a late chronotype. And when you think about that notion, the only source of artificial light that we have at night is moonlight. So this is something I've been interested for several years. We did first a study in non-human primates in the wild that I talked about before, showing that indeed non-human primates have a great modulation of their activity and sleep by moonlight. But talking to the Tobacuom, it was very clear that there's a lot of activity during moonlit nights, and they would tell us about all these anecdotes, right, of things that we're doing during the moonlit night. So one day we decided to formally study this, and this work was pioneered by Leandro Katsuragi, a postdoc in my lab, and also, it's a teamwork with Kaylin, Gideon, and Nacho Spiosas, who's at University of Quilmes, and collaborators at Yale University. So basically, we've been recording. What we did is we went out to the field and recorded longitudinal from the same subjects, actimetry-based sleep for at least one lunar cycle, in some cases for up to 2 lunar successive cycles. And the idea is to see whether there's any modulation of sleep parameters by, or maybe not by the lunar cycle, but at least in synchrony with the lunar cycle. And we recorded from 3 communities: one community that has no access to electricity, one community that lives in a very rural area but has limited access to electricity, like 1 light bulb per household and no street lights, and then a third community that lives in an urban setting with street lights and a highly lit, relatively, environment. And what Leandro did is took all this data from all the communities and first beamed the data according to moon cycles. This is plotted, double plotted, right? But the data is taken longitudinal from each subject. And what you can see is that there's a very striking sinusoidal modulation of sleep duration. This is the duration of sleep, of the bout of sleep during the night, as well as the timing of sleep onset. That is, the onset is earlier. The onset is, yes, earlier when duration is longer, okay? One very interesting aspect of this data is that when we started this study, we were sure that we'd find the longest durations of sleep during the new moon and the shortest durations during the full moon, but that was not the case. In fact, what we saw is that, in fact, the shortest duration of sleep are just before full moon, and they correspond to the latest onsets of sleep, of course. And the same is true for the longest duration of sleep just before full moon, so just before new moon, sorry. So again, the peak and trough does not coincide with the full moon and the new moon. And this is broken up by community. And another interesting aspect is that all three communities had this modulation, although we replicated what we had shown before, that the community that does have access to electricity, which is shown in green, has shorter sleep duration overall, as well as later sleep onsets, but still is heavily modulated or at least synchronized with the lunar cycle. And this was to some extent a surprise because we thought that we'd only see this modulation in the rural community. So one thing that I wanted to show you is the individual data, because of course, when you look at populational data, you don't know whether it's one group of subjects behaving in one way and another group behaving another way. And this is a striking modulation at the individual level by moon cycle. So these are arranged by best fit to worst fit from all three communities, color-coded. And you can see that in some subjects, the modulation of sleep length, sleep duration, and sleep onset are strikingly synchronized with the lunar cycle. And in fact, in some cases, the effect size is of about an hour and a half. That is, a subject can sleep an hour and a half more on the nights just before new moon than he or she sleeps on the nights before full moon. And this is shown just so you get a sense of all the phases in polar plots, now showing all the individuals for the three communities. And again, I want to call your attention that the trough of sleep duration, it's just before the full moon. And coincidentally, of course, the latest onset of sleep, it's just before the full moon as well. So why do we think it happens just before the full moon? And well, it turns out that when you look at the moon cycle, it's very clear why. And in fact, this is what happens in terms of the lunar phase relative to the actual solar night. And what we see is that, in fact, there's a very predictable way in which moonlight is available during the nighttime. And what we see here is that, in fact, the night of full moon, although it's the night in which the moon is brightest, it's the last night in which moonlight is available in the first half of the night. So if you look at the moon cycle, the nights that precede full moon are all the nights that have moonlight on the first half of the night. However, after full moon, the moon starts rising later and later, and you don't have moonlight at the beginning of the night. And in fact, you can see that plotted we recorded this with a very sensitive moonlight photometer, and you can see that on the field sites where we have no electricity, you see this perfectly, where the first half of the night is very bright just before the full moon, but not after the full moon. And what Leandro did is, based on this data from moon cycles, is rebeam the data And he divided the data in 3 sets. The first set is comprised of those nights that have mainly light during the first half of the night, moonlight. Then the second set is the nights that have light on the second half of the night, that is moonlight on the second half of the night. And then the third set is the nights that have no moonlight at all, which are of course around new moon. And then he replotted the data and did this analysis, and the differences are really striking. This is also double plotted data. And what you can see is that, again, sleep duration is modulated. In fact, in all 3 communities, the green community is also highly significant. The blue community is the one without electricity, the one in brown, orange, the one in the rural environment with limited access to electricity. Total sleep, that is the amount of sleep within the bout of sleep, is also heavily modulated by the moon cycle, by moonlit nights specifically. By the way, FML corresponds to those nights that have moonlight on the first half of the night. Interestingly, sleep onset is also heavily synchronized with that lunar light cycle. However, when you look at sleep offset, it's not modulated at all. So in other words, it doesn't matter if you have moonlight on the last half of the night, that will not wake you up. Okay, that's important to keep in mind. And I want you to, and this is just analysis showing that in fact there's no interaction between community and the moonlight modulation, indicating that indeed this is truly present in all 3 communities. One thing that I wanted to highlight is these nights in which we have a lot of moonlight on the first half of the nights. What happens is that the community without electricity approaches the values of the community with free access to electricity. That is the blue community versus the green community. The same for sleep onset. So in other words, on moon nights that have moonlight on the first half of the night, the communities that don't have access to electricity are behaving like the communities that have access to electricity throughout most of the lunar cycle. Excuse me. So what we see here is that in fact, lunar light on the first half of the night is indeed mimicking the effects of artificial light. And this goes back to my notion of chronotype that may have been selected to be active on those nights that did have moonlight during the first half of the night. And when you look at this data is strikingly similar to the data from Christian Kajochen that he published a few years ago in Current Biology, where he retrospectively analyzed a lot of sleep data from a sleep lab in which subjects are poorly sonographically recorded and of course have no access to moonlight information because they are in a lab with closed windows. And yet he found the very same modulation of sleep timing, and also in this case of polysonographic, for instance, non-REM was also correlated with moon phases. So this suggests that in fact, we may have some modulation of our sleep by moon phases, even if we don't have access to the moonlight information. And in fact, when we saw our data on the communities without access to electricity, as well as Christian's data on a sleep lab, we decided to go back to our student data, the data that Gideon has been recording for the last 4 years or so. And we've been all the data, this is data recording from college students in Seattle, a highly urbanized place where the light pollution is always above the threshold of moonlight intensity, at least the moonlight intensity that we recorded during full moon in Argentina, in places where there's no influence of light whatsoever. And yet we still see this striking modulation. This is data from 4 different seasons we've been recording, and this includes up to 600 students. So this is remarkable because here we have students that probably couldn't care less about the moon phases and probably don't even have that information, right? Yet they are displaying these remarkably synchronized sleep timing rhythms with the moon cycle. So this in a way suggests that maybe our sleep is cueing in other physical signals that are correlated with the moon cycle. And in fact, there's these two recent papers, one from David Avery, also from University of Washington, and Tom Ware, that many of you also know, where they showed in a bipolar patient, this is directly related to the subject of this session, we're supposed to talk about health, the impact of light on health. This is a bipolar patient, and this is the sleep length relative to, on successive cycles. These are daily cycles, and black represents sleep. And you can see this striking modulation of these patients, and this patient is from Seattle, by the way. This striking modulation of sleep cycle in synchrony with the moon cycles. But in this case, we see a 15-day modulation, so that sleep gets very short during full moon, but also during new moon. And in fact, you can see this very, very consistently. And then Tom Ware, in another paper, in a couple of papers, he's shown that in fact, in bipolar patients, not only he can see also these sleep modulation. Sleep here is shown in white, in synchrony with the moon phases, but also that the swings in mood from manic to depressive mood, and then back to manic and depressive and so on, are also synchronized with the changes in lunar phases, which is an amazing finding. This only happens to some patients, not all of them. So we actually decided to go back to our data, and I'm sure that many of you that are savvy oscillation observers realize that our data had a dip in the duration and a little peak in the onset of sleep that it's present there, and we noticed that immediately when we started plotting the data. And in this case, what Leandro did was to reanalyze data, in this case with two sinusoids. One that it's a 30-day sinusoid, that would be the synodic lunar cycle, and another one that it's a harmonic of that sinusoid, which is 15 days, so half the duration, and that and then feed this data with these two sinusoids. And what we see is that there's a much better fitting than with the single sinusoid, indicating that there's not only a 30-day component here, but there's also a 15-day component, which is synchronized with the gravitational pull of the moon. And the same happens for the sleep onset. So to summarize a little bit of what I've said so far, I'm getting to my end. So we show that moonlight is associated with a delayed sleep onset and a shorter sleep in general, and that this is only when moonlight is available during the first half of the night. And this is really brings us back to what we do with artificial light today. What we're doing with our artificial light is never using it to wake up at 3 a.m. and start our day of activity, unless you're a shift worker or you need to be at work at 5 a.m. What we're doing is we're extending our evening, and our hypothesis is that indeed what our access, our own manipulation of our light-dark cycle, our access to electricity is doing is emulating the effects of moonlight in our ancestors, because that was the only source of evening light or night light that they had. The moonlight at night is not associated, moonlight in general, is not associated with sleep offset, and that's confirmed in this, what I just said about what we do with artificial light. And then that this modulation is present regardless of the level of urbanization, which is quite striking. As I said, it mimics the effects of artificial light. And I think that the fact that we see this 15-day rhythms of sleep modulation, and also the fact that we still see them in communities where the moonlight cycle cannot be a signal because they're exposed to light levels that are higher than that level, suggests that maybe there's ultimate factors that are related to the moon cycle that are, the ones that have adaptive value, that are probably related to the availability of moonlight, that humans use probably moonlight during the night to be active because it confers some adaptive value. But maybe the proximal factors, those that actually entrain our sleep, if there is true entrainment by the moon phases of our sleep, are probably others, like the gravitational pull of the moon. So what may be that adaptive value of moon cycle? Well, one of the things that comes up when we talk to our Tova communities is that hunting and fishing, when they only relied on hunting gathering, which was only until a couple of generations ago, was always very highly synchronized with moonlit nights. So moonlit nights were always nights in which they would go out and fish and hunt. And then reproductive behaviors is something else that have emerged from many conversations that we have with them. And it turns out that moonlit nights are also correlated with sexual activity. And in fact, there's a lot of mythological stories about this, in support of this. In fact, the moon in the Toba culture is a man that has sexual relationships with women. And in fact, the first period, the first menstrual period is set by the moon phase. And according to the Toba mythology, it will remain in the same phasing for the rest of the life of a woman. And this is in line with recent findings from Charlotte's lab, Charlotte Helfrich-Foster, that showing that there's very strong evidence that in fact, at least in some women and for some periods of their life, the menstrual cycle may be synchronized with moon cycles. So this brings us back to teenagers. And as you know, I've been working heavily with high schoolers and other teenagers. And we know that teenagers have this extreme late chronotype, and we don't know why they are so sensitive to the effects of artificial light. And in fact, I have to tell you that among our populations that we record in the communities without access to electricity, I have a lot of teenagers, and they all show an early chronotype. So one of the hypotheses is that maybe there's adaptive value for teenagers to be specifically responsive to moonlight and maybe isolate themselves for reproduction. And we are now trying to test it to see whether, in fact, teenagers are more sensitive to the effects of moonlight on their sleep than older adults. So that's all I have for you guys. I just like to acknowledge my collaborators, Sachin, Eduardo Fernandez and Claudia Valegia, who are anthropologists at Yale University, and Diego Golombek, who you know from our community, and we do a lot of the wet lab work that I didn't have time to show in his lab, and these are our funding agencies. Thank you very much. I'll stop share this.
Thank you very much for a wonderful talk, Horacio. We'll take questions in the 15 minutes question and answer session. And our next speaker is Sheyum Syed from the University of Miami. Sheyum.
Thank you so much, Christine, for organizing this amazing meeting and for inviting me to share some of our results from recent work. I kind of knew from the title of the section of talks here that I would be included in the midst of a bunch of amazing human studies, and so I made sure that I have Drosophila in my title just to help everybody calibrate their expectations. So I'll be talking about some recent work we have been doing on looking at color preference in Drosophila melanogaster. And as some of you might know, there have been some studies in the past on Drosophila, so this is more of an update than anything else. Okay, so right. So if you don't see the do not take photograph sign, that means you should feel free to take pictures if you want to or need to. Okay, there should be only about 3 or 4 slides that don't have this and that do have this sign, and so those are the only ones where you may not want to take pictures, but the rest are fine for taking pictures. All right. Okay, so color preference, as we all know from our own experiences, color preference depends to a large extent on the context, on what it is that we are trying to do. And so here are some 3 simple examples. If you are out to go for a run or something like that, high level of activity, you probably prefer to be under a blue sky or surrounded by greenery. If you want to rest, maybe the first thing you want to do is block out the blue sky that you had previously. And then similarly, out foraging animals, and we would probably prefer red colors or bright colors, simply because all of these different preferences for different contexts allow us to do something which is to produce or enhance positive emotions of some sort, right? So in each case, when you're running, so this is a set of, let's say, a color palette that we have internally. These are our choices, but when we are out running, we prefer sort of blue or green colors to have some kind of a feeling of exhilaration or fulfillment, right? When we are out foraging, or if animals are out foraging in the forest, they prefer these bright colors, reddish colors, because that's what typically leads them to food, to fruits, vegetables. And so in each case, what we see is that these activities that we want to accomplish determines to a large extent what our preferred colors are towards perhaps this sort of common goal, which is to produce some kind of a positive emotion. Now, these have been studied in psychology for many years, and here are just some papers that you could look up if you want to get deeper into this. But also in these cases, in these simple cases, what we find out is there's also some clear physiological benefit that you can probably get from making these color choices with these certain activities, right? But that's not always so clear cut, at least in us humans. So here's just 2 simple examples where you are now a consumer instead of just a simple active or inactive person. And when it comes to our consumer-related activity, then it turns out that towards the same common goal of maximizing positive emotional feedback, we end up sort of complicating the situation by going for different colors. So, you know, the phone color that you prefer may not be the same one that I prefer, and same for cars or any other consumer items. And so this sort of complication of a very strong emotional component when it comes to picking these items sort of makes this general map between color preference and sort of some kind of an activity. This one-to-one map becomes blurred, at least in humans. So, but this has been studied for a long time. Economists have studied color preference because it's, as you can guess, it's important for the consumer market. But not to be outdone by them, scientists who study simple organisms have also been studying color preference. And here's an example from Christine's favorite organism. in naive monarch butterflies, what's been seen is that if you offer them identical flowers that differ only in their colors, these butterflies will go first towards flowers that are orange in color. Okay. In black-cap birds, if you have a similar, if you do a similar experiment where you offer these birds artificial fruits that now differ only in color again, these birds show a strong preference for red. Okay. And in some trouts, similar studies have been done in terms of where they go or what kind of color environment they want to be in. And in these animals, it's found that trouts, at least juvenile trouts, seem to prefer blue and green over some of the other choices that they're offered. Okay, so not to be outdone by these researchers, so the fruit fly community has also been asking this question and trying to understand the underlying biology of color preference. And so some of the early studies go back to the 1970s, and I just have one example here. There's a bunch of papers that start to talk about color preference or spectral preference around that time. And I'm sort of doing a disservice to that, to those early papers, because I'm really simplifying many different kinds of studies into this one simple geometry. But in essence, for the rest of my talk here, this is going to be, I think, good enough, and which is sort of in very simplified form. Many of these studies were doing the following experiment. They had some kind of a tea maze. They would release fruit flies from the bottom and ask the question of having it, when the fruit flies have light of two different wavelengths, which one do they prefer? Okay, so they release the flies from one end, wait about 30 seconds, maybe up to a minute, and then count the number of flies that are either on the left or on the right. Okay, and so from these early studies, what was seen is that fruit flies, when offered two different wavelengths, they almost always go towards the shorter wavelength. So here I have an example of blue and green. So if you offered the flies blue and green, they would go to blue, most of them. You offered them blue and ultraviolet, they would go to ultraviolet, and so on and so forth. So we knew something about some of the studies in fruit flies, but this is sort of where we started. My lab studies circadian behaviors, and so we have a lot of this, and so do maybe many of you who study fruit fly circadian behavior, where we put, since the time of Benzer at least, we put a fruit fly inside a glass tube of about 6 centimeters with food on one end, and we measured their daily activity. And so if you do this kind of experiment, what you measure is activities, let's say distance moved some per unit time, and what you offer is see as these famous morning and evening burst of activities. So, right, so flies anticipate the rise of sun, and they increase their locomotor activity, and that forms the morning burst. And then similarly, before sunset, they have this evening burst. And so this is the kind of experiment we were doing already. And we thought, well, if we want to ask the flies if they prefer certain color of light, what's the cheapest way to do it? And this is what we came up with. So we just took the old tubes that we had, and we just took some colored filter papers and made 3 sections. We just wrapped up a third of the tube in each of these 3 colors. Okay, so red, green, and blue. And knowing that when we turn on white light from outside, the flies essentially then have 3 rooms, if you may, to choose from, okay, at any time of day. But of course, we had an additional issue, which is we only have food on one end. And so here, what I show here is sort of a couple of sets of tubes that in which we randomize the order of the filter just so that there's no bias towards the food. So the food is at the bottom, and then the order of the filters are arranged. So there are 6 permutations, and so we have sets of 6 tubes in which the order is randomized. Okay. And by using different filters, we also wanted to make sure that the photon flux, or the intensity, so the microwatt per square centimeter through each of these filters is roughly the same. All right. Okay, so this is just a picture of what the experimental setup looked like. This is going to look familiar to most of you in the fly community. So we have infrared light in the bottom for 24-hour illumination. We have white light on the sides to simulate day and then turn them off at night to make night. And we have a camera on top to essentially watch these flies as they go about their daily life. Of course, each fly is in a single tube, so these are solitary flies during the course of the experiment. Okay, so all right, so back in the 1970s, like I showed you, from those studies we kind of knew what to expect. We knew that flies should prefer the shortest wavelength that we gave them, which is blue in our case. Red, by the way, as many of you know, flies don't see very well. So for red in our experiment is considered sort of dim light. So not tied to any specific wavelength, but it's just dim light, a dim white light, if you may. Okay, so given this expectation from the previous experiments, this is what the kind of pattern we expected. We said if we count the number of flies in a whole bunch of tubes and we plotted the fraction of them in each color, so the blue reflects the fraction of flies in blue light or blue zones, green reflects the fraction of flies in the green zones, and so on and so forth. Then at nighttime, we shouldn't see any color specific localization because at nighttime there is no light. But during the day, the flies will be able to make a decision in terms of which room, which zone they want to be in. And so we should expect more flies to be in blue given the previous results. Okay, and this is what was expected, and this is what we saw. Okay, so a big difference. First of all, blue in our experiment we found is the least preferred color. So most flies don't want to be in blue. In fact, they spend the least amount of time in the blue rooms in their tubes. They have a strong preference for green, some preference for dim light, and overall, what you conclude from this kind of a pattern is that the preference for green is modulated in time. So there is a strong preference for green around 2.5 hours after lights turn on. This preference for green dips in the middle of the day with a simultaneous increase for preference for dim light. Okay. And then this preference for green resurges towards the end of the day again for a second time. Again, and all this time, the preference for blue stays low. Okay. And so for most of the talk, I will be focusing on sort of the green and the dim, and hopefully I'll have some time to just quickly summarize the blue result. Okay, so here's a short movie. Let me just sort of give you the landmarks a little bit. So on the left side, I have the color preference on top, okay, plotted in every 3 minutes. If I looks a little jaggedy. And the bottom is the very familiar locomotor activity pattern. So you can see the position of the green peaks with reference to the locomotor peaks. Okay, and here I have a movie, and the arrows are going to be somewhat synchronized to each other and somewhat synchronized to the movie. but it's not perfectly synchronized. Okay, and the light is off now. We will start when it's sort of middle of the night, and then slowly go through the sunrise and sort of stop 4 hours after sunrise. So, okay, here we go. So it's still nighttime. Morning activity starts, and you can see the flies throwing themselves around like crazy, right? With total disregard for color of light. Light turns on, of course. Now they start to settle down, lower their locomotor activity, and settle down into these various compartments. As you can see, most of the flies are in green, but some are in some of the other colors. I'm just going to run it one more time. So nighttime, light turns on, locomotor activity will slow down soon. And then the soul slow down. Okay, and here's another movie. So all the data is essentially in the movie. So here we focus on the middle of the day, sort of 6 hours plus minus a couple of hours after lights turn on. So most of them are in green, but then they switch. Some of them switch to red. Some of them stay in green. Okay. All right. So yeah, if you are unable to reconstruct the data from the movies, here it is. They prefer green early morning and late afternoon, prefer dim light and green light middle of the day, and they do this for days at a time. So here's about a week of data. I think we have tested maybe for about 2 weeks at most, and this kind of a pattern continues on at least for 2 weeks. Okay, so right, so this kind of data set, of course, looks like looks a lot like sort of the bimodal locomotor patterns that I was just showing you, and sort of this made us wonder if circadian clock is involved, of course. And so one of the first things we did, we looked at clock mutants. So for those of you who are unfamiliar with the fly clock, here's a full cartoon from an old paper review by Paul Taggart and Michael Nitterbach. And so just to remind you of some of the important clock genes. So if we look at per null, tim null, and psych null, they all show more or less the same phenotype, and which is sort of the modulation between green and dim light is absent when you knock out the clock. So that very clearly suggested that at least the temporal modulation that we see in color preference is somehow related to this internal clock. Okay, so if we, so this is a sign I'm going to use at least one more time. This is the sign for internal clock, and this is a sign for external cycle. If the two are matched in wild type flies, so put them in 12-12 cycles, we see this familiar pattern now, and just in reference, you can see also the morning peak followed by the green preference peak, daytime siesta, equal distribution between, or roughly equal between green and red, evening activity peak, and so on and so forth. Okay, so if you do this for different permutants, a long, short, this is a short, wild type, and per long, where you match the internal and the external cycles, they look wild type-ish, by which I mean there are these bimodal peaks in green preference, midday, split between dim light and green. So if you match that for internal, external, for per short, for wild type, or per long, that pattern stays the same. However, if you now tune the internal and external clocks, or rhythms or cycles, where now you have per short in its matched cycle, but wild type in a short external cycle, the wild type pattern disappears. So you see that the sort of abnormal patterns in locomotor activity, and similarly abnormal patterns in the color preference. Okay, and this is true for all of these mutants. Okay, okay, I'm going to speed up a little bit. I have only a few minutes left, and so something that we started to do recently is we said a couple of years ago we published this paper. We were showing how one can, on a circadian time scale, monitor multiple fly behaviors, including grooming, which is sort of a subtle fly behavior. And we thought, well, we could perhaps look at what flies are doing in these different colors. And this is sort of sketched out here. These are very preliminary data, but on top of the color preference, in the bottom are some of the behaviors that we can track in these different colors as they go in and out of the different rooms. And here's an example. Oops, sorry. Right. And so this is the quantification of some of that data, and you can see that flies do have a certain preference for where they want to do what, right? So this kind of picture is emerging, similar to what I was talking about for human behavior, right? So flies don't do the same things in the same colors. Okay. So back to the middle of the day, I have blocked out all of the flies that were in different tubes, except for the ones that are in tubes with blue next to the food. Okay, so except for these two. And you notice that the flies where they have food next to the blue park themselves in the adjacent room, and they only make these very quick trips to eat in the middle of the day. So this is middle of the day. It's kind of, okay. And this disappears in certain mutants, including the TRPA1 mutant. And we also discovered that if we lower temperature, the wild type flies start to resemble the TRPA1 mutant. And this alone would not really tell us that there is some sort of an interaction between color preference and temperature preference, but we have some more interesting data that you can look at if you visit Stan's poster later today, where he talks about this kind of an intersection between color preference and temperature preference that we have started to explore. Okay, so I haven't told you exactly how the color light enters the fly body. Not too surprisingly, eyes are involved. Here's just a refresher for the photoreceptor system that flies have. Flies have 6 photoreceptor cells that surround 2 photoreceptor cells, R7 and R8 in the middle. And when we knock out the fly visual system in multiple ways, I guess not too surprisingly, the preference for green versus red versus blue is affected differentially. So the one for green and red disappears while blue remains intact. Of course, the blue remaining intact is surprising, but this is not too surprising, the green versus red. Now, what was surprising is that this color-specific behavior seems to be mediated by a broadband rhodopsin, which is rhodopsin 1, while flies have all of these color-specific photoreceptors, which don't seem to be involved in this color-specific behavior. Okay, especially rhodopsin 6, which is green sensitive. All right. So I've showed you multiple cases where the green-red is affected, but you may not have noticed that the blue avoidance is unaffected in all of these cases. So clock mutants, TRPA1 mutants, visual mutants, and so forth. So that led us to think that this part, at least the green dim light preference, is modulated through this kind of a pathway, which is independent of blue. And for blue, we searched long and hard and ultimately realized that if we manipulate the peripheral neurons in the fly body and the fly abdomen, which in larvae have been shown by the Jan lab early on to mediate blue avoidance, those same multidendritic neurons seem to be involved in the blue avoidance behavior that we observe in our experiments. Okay, so this is not the multidendritic neurons silenced. And interestingly as well, rhodopsin 7, which is the newest rhodopsin in the field now, still some controversy involving it, this rhodopsin seems to be the one that's mediating this avoidance behavior. Okay, so that's about it. Here are all the people that we are thankful to. Stan did bulk of the work, helped by his brother Andre, James in our biology department, a close collaborator. And then we talked to a bunch of colleagues and friends, and we were lucky to get funding from the National Science Foundation. And so I will stop right there. Thank you.
Thank you for a wonderful talk. So our next speaker is Diego Fernandez from the National Institute of Mental Health. Diego, the floor is yours.
Thank you very much. And thanks everyone for being here. It's an honor to be part of this session, to be online today. I want to show you some data, recent studies from the Hatar lab. And basically, or briefly, we will discuss some of the light effects on mood, learning, and metabolism. I'd like to thank the organizers and also Samer Hatar, my mentor, and all the people in the lab for their support. In our lab, we are interested in light and how different circuits can drive the direct effect of light on different aspects of animal behavior. We work with mice. So as you may know, in mammals, the retina is the organ that exclusively encodes light information to the brain. In this schematic is a foundational discovery from Monica Hall. You can see the different layers in the retina. This is a beautiful description of photoreceptors that send light information through bipolar and interneurons to these projection neurons. These cells send projections to multiple areas in the brain. They are considered ganglion cells, retinal ganglion cells. And of course, as you can imagine, light is crucial for vision. So most of these cells project to areas that are involved in conscious vision, image detection, movement detection, different aspects of vision, visual physiology. But we're also interested, and we know that light is also important for the alignment of the internal timing with the external light-dark cycle. And this is a process known as photoentrainment, and is mediated by the central pacemaker of the suprachiasmatic nucleus. And what is remarkable is that the innervation from the retina is exclusively from a subset of melanopsin cells. These are retinal ganglion cells that express photopigment called melanopsin and were discovered in 2002, around 20 years ago, and were originally described as an homogeneous population of cells, but we know now that there are in fact different subtypes. Here I'm showing a model that represents the 6 subtypes of iPRGCs that we know today. And we have a good characterization of their morphology, their electrical properties, and we are starting to understand the connectivity with the brain. But if we combine all these cells together, we have this beautiful map of innervation describing the complexity of the retinal brain connection. And as you can see, there are targets that are part of the midbrain, dorsal thalamus, of course, the hypothalamus. And a ton of these areas have a role in functions that are not directly related to circadian rhythms. So this suggests that light can have direct effect on these functions that are, of course, subconscious functions associated with mood and learning functions. And this becomes particularly relevant when we think in human disorders. Some of the disorders we know are caused by alterations in light And nowadays we are all exposed to artificial light conditions, which has become a major form of pollution that has a negative impact on human health, affecting circadian functions, sleep, and affective behavior. And some of these alterations are caused by extreme lighting conditions, as experienced when we traveled across different time zones, particularly repetitive travels across time zones, and especially in people, by people experiencing these shift work schedules. And this is particularly relevant nowadays, and it's something that we should consider for the next or for the future societies that we are planning to design, and it becomes critically relevant now. So in all these cases, and in most neurological disorders, it has been associated with changes in clock chains, leading to this linear relationship where abnormal light conditions disrupt circadian functions and sleep architecture, thereby causing mood deficits and learning deficits. So this linear connection made the assumption, or lead to the assumption, that most of the alterations are caused by altering the circadian rhythms or the sleep architecture, indicating that this is the secondary effect of light. But recent data have challenged this view and showed that this could be even more complicated. And in fact, light could have a direct impact on mood without causing disruption of these rhythmic properties. So one really important study was published in 2012 from the Hattal lab, and they showed that mice exposed to this ultradian light cycle, the T7 cycle that present short light pulses and dark pulses 3.5 hours each at both active and inactive phases of animals, do not cause severe disruption of circadian activity, as well as no alterations in clock chain expression. However, these animals showed sign of depression and learning deficits. And in this study, they showed also that IPRGCs are crucial for the retinal circuits driving this direct effect on mood. So we wanted to elucidate the mechanisms, the circuits, the retinal brain circuits that could drive these direct effects. And we explored different targets in the brain, different areas innervated by IPRGCs. And we discovered a region of the dorsal thalamus that fulfilled with all the requirements to be a candidate driving the direct effect of light on mood. First, we found that this region of the dorsal thalamus, termed perihabenular area, receives dense innervation from the retina. You can see these coronal sections as well as dorsal views of the brain. In green, you can see all the axons from the retina. It's densely innervated. We also discovered that these are exclusively IPRGCs. And interestingly, it shows, or it displays robust neuronal activation in responses to environmental light changes. This is a CFOS induction. You can see here the dramatic induction of CFOS when we give light pulses to animals. And it's very interesting to know that when animals were exposed to the ultradian light cycle, the T7 cycle, they still showed this strong induction of CFOS, suggesting that every time that the animal is exposed to these abnormal light conditions, these cells are being activated. And remarkable, PHB is integrated in a distinctive circuit with mood regulating centers. Using a battery of tracing strategies, we found that these PHB neurons project to prefrontal cortex, dorsal striatum, as well as the nucleus accumbens. So after this characterization of the circuitry, we decided to explore and manipulate the PHB circuits to see how this could impact on mood behaviors in mice. And we use strategies to chronically activate and silence the cells. I'm showing you here one example where we use a 2-virus strategy to induce the expression of the DREAD receptor exclusively in the cells of the PHB that project to prefrontal cortex. This is the excitatory version of the DREAD, so we can manipulate the cells and cause chronic activation of these neurons. It's important to mention that animals were all time in normal light conditions, T24 cycle. And 4 weeks after the injections, we treated the animal with CNO, which caused the chronic activation of these receptors. The idea is during 2 weeks, animals were drinking water with CNO, which caused this chronic and abnormal activation of these neurons, exclusively these neurons. This is bilateral injections, by the way. So what we found at the end of this treatment is that animals showed severe deficits in mood-related behaviors. These are 2 examples, the tail suspension and forcing test. These are classic examples of mood-related behaviors used in the depression field. And basically, we are measuring here the time that the animal are trying to escape, or on the other hand, the immobility time, which is considered a sign of helplessness associated, of course, with clinical or human depression. So in both cases, you can see that controlled animals try to escape most of the time, but these dread-treated animals showed an increase in the immobility time, which is a clear sign of alterations in mood. And interestingly, the same conditions, the same treatment, do not affect learning in these animals. I'm showing you one example. This is the novel object recognition test, where animals are exposed first to an arena with two identical objects, and an hour later are re-exposed to the same place, but now one of the objects is replaced by a new one. And as you can see, both groups have a clear preference for the novel object. And in fact, we found that a parallel circuit is mediating this direct effect of light on learning. As I told you before, when we expose animals to the T7 cycle, we found both learning and mood deficits. So this result revealed that there are two parallel pathways that are involved, in one hand, retinal projections to the SCN, and on the other side, retinal projections to the PHB to directly affect mood and learning in mice. We still don't know the mechanisms or the circuit connecting the SCN with the hippocampus or the learning functions, but we know definitely that this is mediated by exclusive population of ganglion cells that project to the SCN and use a non-canonical function of the SCN. We found this because all these animals showed no disruption of rhythms. However, we propose here that these cells can still relay information to affect learning functions. And in parallel, we uncovered this region or this connection of the retina with PHB affecting mood regulating centers that is exclusively involved in driving the direct effect of light on mood. And I invite you all to visit later today a poster from Tenley, where she will further explain and describe new data characterizing this thalamic region associated with mood control. So together, these results reveal that abnormal light conditions have a direct impact on mood and learning. But in a broader view, we could also think that irregular light exposure could provide conflicting information between environmental light and non-phoric cues. So to understand whether this conflicted information could have a negative impact on physiology, we decided to take a step back and explore the potential mechanisms underlying the integration of multiple time cues to drive a coherent circadian output or a coherent behavioral output. So we decided to explore two of the strongest time cues in nature and the mechanism that can or could potentially integrate these two signals. We studied light and restricted access to food, okay? So our prediction was, if we removed all the retinal input to the brain, this would cause a remodeling and potentially a strengthening of entrainment to non-phoric cues, such as time-restricted feeding. And we were inspired by data or research that focused on the superior colliculus. This is potentially the center that integrates most of the external signals from the environment, not only the retina, but sensory information, visual, auditory, motor information. And what is remarkable is that there is a critical window during which the ablation of retinal input caused a complete remodeling of the entire circuitry, leading to a strengthening of non-phoric modalities. So with this in mind, we decided to explore, this is, one example where you could see all the remodeling caused by the enucleation in young animals. So we used a mouse line that was generated in our lab, where we expressed the diphtheria toxin, DTA, under the control of the melanopsin promoter. So as you could see in this example, these animals do not have IPRGC innervation starting from early in development. So this is a great model to study the ablation of IPRGC exclusively. These animals have normal innervation from conventional ganglion cells. And to evaluate non-phoric entrainment, we use a classic time-restricted feeding paradigm. It has been extensively used in our field and characterized in the field. There are multiple papers that describe and characterize the behavioral and physiological changes associated with this paradigm. In this example, you can see that when animals are exposed to this restricted feeding condition where they have access to food for 7 to 5 hours, depending on the model, you can see that after a couple of days, they start showing this robust anticipation, which is a clear sign that the animals are now entrained to this non-photic cue. And it's also showing that something that happened in the past is affecting, the behavior and future behavior. So we decided to keep animals under constant darkness, a similar paradigm, but now without light information. The idea here is that the time-restricted access to food constitutes the only recurrent time cue for these animals. So in this, I will show you some actograms with this pattern. Every time you see orange, it means that they have access to food. In gray, it represents no access to food. And green is the area where we expect to see the anticipation, similar to what I show you here. So this is the paradigm we use. Animals are single-housed, and we are using feeders to control the amount of food and when they're getting the food. And this is an example of an actogram of a controlled and a DTA animals without this IPRCC innervation. You can see that in both cases, under ad libitum conditions, they have this beautiful free running period, and then we expose them to restriction, and they showed similar responses. As expected, control animals show this robust anticipation. You can see after a couple of days, they have this robust anticipation. It's about 3 hours before food access. And when we measured the DTA's animals, we were shocked to see that these animals do not show any sign of anticipation, or is really, really weak, it was honestly a surprise for us. It was exactly the opposite to what we were expecting. And in fact, if you see this animal, you can see the free running in ad libitum conditions. There is no affected at all by the transition to the time restricted feeding. This is the quantification. Most of the animals show a similar response. Of course, they are in free running, so there is a big variability between animals. I invite you all to see the paper where we have all the actograms from different animals, and the average of the activity before the access to food is shown here. You can see that all the animals show significant reduction in the anticipatory responses to this paradigm, and this is the average of, or the percentage of activity 3 hours before food access. So we then wanted to explore potential circuits and mechanisms underlying these behavioral responses. And we reasoned that this should be an area innervated by IPRCCs. It has been shown that there are multiple areas in the brain and outside in the periphery that show changes in neuronal activity caused by this time-restricted feeding. But these alterations observed in DTA suggested that it should be a target of IPRCC. And also, we assume that it should be a region driving non-photic signals to circadian centers. So I decided to explore the IGL as a good candidate that fulfills all these requirements. The IGL, the intergeniculate leaflet of the thalamus, is an area that received dense innervation from IPRCCs and is connected with the SCN. It's connected with multiple brain areas, but mostly project to the SCN. This is a beautiful connection, since IPRCCs are also innervating the SCN. So to evaluate if there is any correlation between the function of these neurons in the IGL with the protocol that we used, we exposed animals again to the time-restricted feeding paradigm. And after 21 days, we collected samples from animals right before the access to food, okay? A couple of minutes before receiving the food, during this anticipatory period, we collected brain samples and we measured the CFOS induction as a readout of neuronal activation. As controls, we kept animals under ad libitum conditions. And again, let me remind you that this is all under constant darkness. Okay, so control animals showed a substantial and significant induction of CFOS, reflecting that these neurons are associated with the responses, or potentially associated with the responses we observed previous to the food access. But what we found when we exposed the DTase animals, the animals without IPRGCs, to the same paradigm, is that there are no differences between control conditions and this time-restricted feeding, which is the same or similar to what we found with the behavioral responses. The IGL contains MPY neurons that projects to the SCN, and neuropeptide Y has been broadly associated with feeding responses and is expressed in brain areas associated with feeding and hunger. So we measured the levels of MPY in the IGL of both control and OPM4 DTase animals, and we found a significant reduction in the expression of MPY. These are samples collected at CT14, in both cases animals that were in constant darkness for a couple of days. And we also measured the projections to the SCN, and we found a significant reduction, indicating that not just the SCN, but potentially the connection with the SCN could be involved in driving these behavioral alterations. So all these results, these behavioral and morphological results, indicate that IPRGCs are playing a crucial role in the assembly, or in some way affecting the functional connectivity of these neurons between IGL and SCN. So to evaluate whether there is a critical window during which IPRGCs affect these functions, we decided to use wild-type animals that were enucleated at different times during development. So we enucleated animals excuse me, at P0, P40, and P90. Sorry. So these animals were all enucleated early in development or late during adulthood. And a couple of months later, we analyzed the connectivity of the IGL with the SCN, particularly through this MPY expression. Good. And we found that P0 and P40 are similar to what we found with animals without IPRGCs from early during development. However, animals that were enucleated by P90 do not show these alterations, suggesting that there is a critical window by which IPRGCs influence the assembly of these circuits. And we found a remarkable correlation with the behavior. As you can see in these representative actograms, animals that were enucleated at P0, P40, and when we're exposed as adults to this paradigm showed very weak anticipation, whereas animals that were enucleated at P90 show a normal response, similar to controlled animals. This is particularly important because the data reveal that IPRGCs play a role during early development. There is a time window before P90. We don't know the details, but it should be early in development. However, these are all adult animals. So we wanted to explore whether this circuit is playing a role, an active role in adults. And for that, we use MPY animals where we could silence the function of these cells. So briefly, we use a strategy to induce tetanus toxin expression in these MPY-CRE positive neurons. We injected the virus into the IGL, causing the expression of tetanus toxin and therefore the silencing of the release of these terminals. After 4 weeks, after injections, animals were exposed to the restriction paradigm. And as you can see in these representative actograms, these animals showed really, really weak anticipation, and it's all quantified here. Obviously, these cells have multiple projections, but it's enough to show that these cells are sufficient in these responses, to drive these responses. So to further characterize the connectivity of IGL to SCN and its roles in these functions, we use a similar strategy, but now it's an optogenetic control of the terminals from these MPY neurons from the IGL projecting to SCN. So we implanted cannulas and fibers on top of the SCN in animals that were injected with a virus that express this ARCH-T. Is a optogenetic strategy to silence the cells, acute silencing that is completely reversible. So after the injections, we place animals in the same restricted paradigm. And after 2 weeks, where we confirmed a robust anticipation, robust food anticipatory response, we measured the activity and applied this inhibitory protocol. Briefly, we measure locomotor activity for 3 hours before access to food. So the first hour will be our control. This is when we confirm that the animals are in fact showing a robust anticipation. And starting 2 hours before access to food, we apply these pulses of inhibition of 20 minutes with intervals of 20 minutes. So this is what we found. Animals that show a robust response during these first hours now show a significant reduction when we silence the terminals, demonstrating that the projections from the IGL to the SCN are required for entrainment to time-restricted feeding.
This is so cool.
So it has been previously shown that light can affect a light entrainable oscillator that is part of the central pacemaker of the SCN. And similarly, it was proposed that there is a food entrainable oscillator that controls these anticipatory responses to time feeding. Here we propose that IGL is a node of connection between these two oscillators, and during time-restricted feeding, inhibitory signals from the IGL silence neurons in the SCN, leading or allowing field to take control of the activity of animals. And if we combine all these results, and to sum up, the results and this line of research contribute to the understanding of how independent time cues are processed by different brain circuits, as well as how these coded signals are then integrated to drive a coherent behavioral output. With that, I'd like to thank all the collaborators, all our great collaborators, Alfredo Kirgut, David Burson, and Mario Penso at NIMH. Some of the results I showed you were done in collaboration with them. I would also like to thank the people in the Mousetrail Lab at Hopkins, as well as the people at NIMH. I want to highlight the work of Ruchi Kamal. She was essential in the data I showed you before about feeding and the anticipatory responses. And there are some beautiful posters today in the afternoon, tomorrow. I invite you all to go and see this new exciting data. And finally, I'd like to thank my mentor and friend, Samer Hattar. Thank you very much.
All right, thank you, Diego. I think we're gonna be moving towards the question and answer session right now. We only have 7 minutes left before the end of the symposium. So everybody, I saw that some of you have already started to answer questions through the chat. There are a few questions. The first one, maybe we can do one question each within the time allotted. So there's one question for Claudia. Did you see seasonal differences and gender-specific differences in your studies?
Oh, thank you for this question, and thank you for the nice session. Actually, we don't believe there are seasonal differences in this population because they don't change through the year, this location. The temperature is the same, the sunlight is the same, they are at the 10 degree of latitude. The only difference is what they call winter. They are more rain, is what we call rain season. So we believe the behavior would not change, but we didn't compare with this two seasons. And we had only males in our samples. Thank you.
Okay, there is another question for Horacio from Teal, asking, your data suggests that chronotype distribution in pre-industrial population should be wider in places with less cloud cover, since it would be more mimicking more artificial light.
Yeah, that's a great point, and it's something that could be tested. One thing, however, that I need to point out, that even on cloudy days, moonlight, and we actually measured this, we were worried because in one of the moon cycles that we were measuring moonlight intensity with our photometer, it got very cloudy throughout the moonlit nights, but we still were able to pick up that light intensity. So even when you don't see the moonlight, there's a big glow that you can actually, that it's measurable, and maybe we are sensitive to that.
Okay, there's another question for you, Horacio. Is there an effect of quarter moon on sleep onset and duration?
Sorry,
Is there an effect of quarter moon on the sleep onset and duration?
Well, it's intermediate, so you could say there is an effect because it's intermediate between the peak and the trough, right? So yeah, I guess you could say there is an effect.
Okay, there are other questions. So most of the question for, sorry, this is complicated to just read everything and sort everything out. So for Diego, Lorena Blitz is asking whether this IP, RGC, IGL loop work with other non-photic cues like exercise as well.
Okay, yeah, that's a great question. It's an interesting possibility. We don't know whether other non-photic cues would use the same system. It's not easy to measure since anticipation to food is potentially the only cue that gives rise to this strong anticipation. So it could be really nice to have a model where we can see clear signs of anticipation and evaluate whether this circuit is also involved. At the same time, there are multiple areas projecting to SCN. So it is also possible that multiple areas in the brain could act as nodes of connections between potential oscillators and the central pacemaker.
Yeah, I guess related to that, there's another question for you. Are there outputs from the SCN that also reach the PHP? Or are these segregated pathways?
We couldn't find connections between PHP and the SCN. When we use the rabies virus strategy, we haven't seen cells in the SCN connecting SCN neurons with the PHP. However, it could be an indirect pathway.
Could it be polysynaptic?
Yes, could be connected through another area that it's both SCN and PHP. Yes.
So, Sheyum, is there any question that you would like to address? I've seen that you've answered a lot of them in writing. Is there anyone that you would like to address to the public?
No, yeah, I've been working diligently on the question answers section. Yeah, I think I've answered almost all of them. I don't see any that I have not yet. Okay,
so maybe I will continue on a few opens before the session is over. We have just a couple of minutes. Yeah. There was a question or a comment to both Claudia and Horacio, asking whether differences in chronotype, this is a comment, differences in chronotype have long been postulated from theoretical and experimental work to depend on three factors: genes, age, and light. And under strong Zeitgebers, you expect a narrow distribution of chronotype. That is exactly what this hunter-gatherer society data show, but also comparison between urban and rural communities. So I guess this was not a question, but rather.
Yeah, we agree. You know, both Claudia and I agree with that.
Yeah, yeah.
Okay, so we have 1 minute left. I think we will just close the session. Before doing that, I would like all of us to really thank these four speakers for a fantastic session. And if I suspect the question will be sent to you guys, so you will be able to continue answer them if there is anything that hasn't been answered. So please join me in congratulating Claudia, Horacio, Sheyum, and Diego for a fantastic job today.